Here is a question that doesn't get asked enough in the pharma industry. How do you prove that your peptide is the same as the innovators? Not just similar, not just bioequivalent, but actually the same. Because if you get that wrong, the FDA will tell you, and that conversation is expensive. Hi, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, Canada. Welcome at Resolve Mass, and we bring deep scientific expertise in advanced analytical characterization and complex molecule development to accelerate your IND and ANDA programs. Let's start with why this even matters. Because if you're working on a generic peptide, whether it's a cyclic peptide, a disulfide bridge molecule, or something more complex, you probably already realize that bioequivalence studies alone won't get you across the finish line. Regulatory agencies, particularly the FDA and Health Canada, require something more rigorous. A comprehensive demonstration that your generic and the reference listed drugs share the same primary structure, the same higher order conformation, a comparable impurity profile, and similar stability behavior. That's not a single test, that's a whole program. And here is where a lot of teams run into trouble. They underestimate how nuanced this reality is. Even a single amino acid substitution, a scrambled disulfide bond, or slightly different impurity fingerprint, any of those things can raise a red flag in a regulatory review. So let me walk you through how you build the evidence that answers those questions convincingly. Everything begins with confirming primary structure. And I know that sounds basic, but that devil is very much in the details here. The workhorse for this is high resolution mass spectrometry, HRMS. It gives you exact molecular mass to a level of precision that leaves very little room for ambiguity. If there is an amino acid substitution, even a conservative one, HRMS will catch it. But mass alone doesn't tell you the sequence. That's where tandem mass spectrometry or MSMS comes in. You fragment the molecule, and from those fragments you read the sequence like chapters of a book in order with confidence. Truncations, unexpected extensions, they show up clearly. And then there is a peptide mapping by LC-MSMS, which is probably the most information-rich technique in the toolkit. When done right, it gives you near complete sequence coverage and simultaneously flags modifications, things like deamidation, oxidation, or acetylation that you might never catch with a simpler approach. The practical takeaway here: don't rely on a single mass spectrometry experiment, run the full suite. Each experiment answers a slightly different question, and together they build an airtight case. Now here is where it gets genuinely challenging. That's the higher order structure, the harder part. Higher order structure analysis and the conformation, folding, disulfide bonds. Think about this in two ways. Like, two peptides can have identical amino acid sequences and still behave completely differently, because if they have disulfide bonds that are paired incorrectly, or if the molecule folds into a different conformation, that's not a hypothetical risk, it's something we see in practice. For disulfide bond mapping, we are looking at partial reduction strategies paired with mass spectrometry, identifying exactly which cysteine pairs with which and confirming that your generic matches the reference product's connectivity. Secondary structure analysis using techniques like circular dichromism adds another dimension. It tells you whether the molecule's overall folding pattern is consistent. This is an area where having deep hands-on experience really matters. Some molecules are beautifully cooperative. Others, especially those with multiple disulfide bonds or non-standard connectivity, requires custom workflows and careful method development. There is no one size fits for all kind of protocol. So impurity profiling. If primary structure is the foundation, impurity profiling is arguably where regulatory scrutiny is most intersite now. Think about what's in your sample beyond the main peptide. You have process-related impurities from synthesis, degradation products, sequence variants, aggregates, and FDA wants to know not just what they are, but how they compare to the reference product. Reverse phase HPLC remains the standard purity method. It's robust, it's well understood, and it's what reviewers expect to see. UPLC gives you high resolution separation when peaks are closely spaced and you need to pull apart the things that traditional HPLC can't resolve. And HRMS, combining high resolution mass spec with chromatographic separation, lets you identify low level unknown with confidence. One thing we have found at Resolve Mass in our work with complex generic peptides, co-eluting impurities are among the most common challenges. You think you are seeing a single peak, and it turns out that there are two overlapping species, one of which is a sequence variant you didn't know was there. Orthogonal chromatographic approaches with different stationary phases or mobile phase conditions are essential for catching this. Beyond purity, you are also doing physicochemical comparisons, retention time alignment, isoelectric point, hydrophobicity profiling, etc. These aren't just box checking exercises, they are part of painting a complete picture of a molecular sequence. Aggregation and stability, proving the sameness over time. Here is a question worth sitting with. What happens to your peptide under stress? Aggregation and stability studies are the part of sameness testing that looks forward. They are asking, does this molecule behave the same as the reference, not just today but under challenging conditions? Size exclusion chromatography with MALS, multi-angle light scattering, is the gold standard for aggregate quantification and characterization. Dynamic light scattering adds complementary data on particle size distribution. Together, they tell you whether your molecule is prone to aggregating in ways that differ from reference product. And from immunogenicity standpoint, that matters enormously. Force degradation studies, acid and base hydrolysis, oxidative stress and thermal stress, and photolytic exposures are designed to break the molecule intentionally and then compare how it breaks relative to the reference. Do you see same degradation products in similar proportions through similar pathways? If the answer is yes, you have just added another layer of evidence that these two molecules are fundamentally the same. Let me bring this all together because this is really the key concept that regulators are looking for. No single analytical technique is sufficient for peptide sameness demonstration. That's not my opinion, it's the regulatory expectation. The FDA wants to see what they call a totality of evidence approach. meaning multiple independent methods, each confirming the same attributes from the different angles. So molecular weight confirmed by HRMS and MALDI-TOF, sequence confirmed by LC-MS and peptide mapping, disulfide bonds confirmed by mass spectrometry mapping and NMR, impurities confirmed by RPHPLC and LC-HRMS. This is what we call an orthogonal analytical strategy, and at Resolve Mass, it's backed into every sameness testing program that we run. Because when you show a regulator that two completely independent methods converge to the same answer, you dramatically reduce the chance of a deficiency later. Regulatory challenges. Now let me be real with you for a moment, because this work is hard and I don't want to sugarcoat it. The common challenges we see teams struggling with: low-level sequence variants that sit right at the detection limit, correlating impurities that need orthogonal methods to resolve, complex disulfide patterns in molecules that don't behave nicely during reduction, process-related impurities that don't have a clean reference in spectral databases, and then there is an RLD comparison problem. The reference listed drug comes in a formulated state and you need to strip away excipients, normalize concentrations, and still generate data that's directly comparable. That requires careful method development and a solid scientific justification. Even one of these challenges is solvable, but they require experience, the right instrumentation, expensive methodological approach, and cutting corners at this stage is where submission go sideways. So what's the takeaway from all this discussion throughout? Peptide sameness study isn't just an analytical checkbox, it's the scientific narrative that tells regulators we understand this molecule, we have characterized it thoroughly, and we can demonstrate with multiple independent lines of evidence that our generic is the same as the reference. That narrative needs to cover primary structure, higher order conformation, impurity profiling, and stability, all supported by orthogonal methods, all scientifically justified. If you are at any stage of that journey, whether you are just starting to plan your analytical strategy or you have hit a wall with a specific technical challenge, I'd genuinely encourage you to have that conversation with an experienced lab that lives and breathes this work. Because the question isn't whether regulators will ask hard questions about your peptide. They will, they'll surely ask. The question is whether your data package is ready to answer them or not. Thank you so much for watching this session, and see you in the next one. Goodbye for now.